almost done, inshallah. Okay, so I think we should start. Hello, everyone. My name is Ghayed Alwassiah. I'm a second-year medical student. And today I'm going to be giving you guys the PAL session on the urea cycle. It's a really simple lecture, so inshallah you guys will find that it is. It's also really fun in terms of like the genetic deficiencies, and we'll get to that in the end. But inshallah you guys will find that it's simple. So okay, should we go ahead? Okay, so first we'll discuss where the urea cycle happens, you know, what we need for the urea cycle to proceed. And then of course, we'll talk about the steps in detail. So there are 5 steps. It's a very short cycle compared to other things that you might have taken. And then we'll talk about how we regulate the cycle. So the important steps, the important enzymes, and so on and so forth. And then the fun part, which is the genetic deficiencies. And I also included some practice questions for you guys. So we can go through those together, and I can show you how to differentiate between different diseases and how to answer questions. Okay, so why do we need a urea cycle to begin with? So the urea cycle is pretty much the body's way of getting rid of ammonia. And ammonia is a molecule that can be very toxic if it builds up in the blood. So there has to be a way that the body can get rid of ammonia. And this is how it does it. So we put ammonia through the urea cycle and we end up with urea, right? So from the name urea cycle, the final product is going to be urea. And that's how the body excretes the ammonia in the form of urea. Now, like we said, ammonia is toxic, right? So ammonia can't be in the blood. So in order to transport ammonia to where the urea cycle actually happens, we need a carrier that is non-toxic. Okay, so let's say we have ammonia in other tissues of the body, right? How do we bring it to the site where the urea cycle takes place? We will convert it into something that is non-toxic, and that something is glutamine. Okay, so this could come as a question for you guys. So glutamine is the non-toxic carrier of ammonia. So it's the way that we can transport our ammonia into the liver where the urea cycle takes place. And so to convert our ammonia into glutamine, we have this reaction over here where we get our ammonia with glutamate to make glutamine. And then this glutamine can go through the blood and to the liver and be incorporated into the urea cycle. Okay, so again, to repeat, the main takeaway from this slide is that glutamine is the non-toxic carrier of ammonia. So like we mentioned, the urea cycle will take place in the liver. So from start to end, it's all in the liver within the hepatocytes, which are our liver cells. So in the liver cell, we'll have 2 locations. It can either take place in the matrix of the mitochondria or in the cytoplasm. So the first 2 steps will actually take place in the matrix, and then the rest of the cycle will take place in the cytoplasm. And we'll look at that later on as well. So for the urea cycle to proceed, we need 3 specific atoms, okay? So it's 2 nitrogen atoms and then 1 carbon atom. So this 1 nitrogen atom of urea will come from aspartate. The other one comes from ammonium, and then the carbon atom comes from our carbon dioxide. Another thing to note is that in the urea cycle, we have a specific molecule known as ornithine. And ornithine is special because it acts as a carrier for all these atoms, right? So it carries these atoms that we need for the urea cycle. And we'll keep repeating this as we go on. Just this is kind of like a brief introduction. So this is like a little visual representation, right? So for the urea cycle to proceed, we need the nitrogen atom from ammonium, we need the carbon atom from carbon dioxide, and we also need the nitrogen atom from aspartate. Okay, so this is the urea cycle. This is the overall reaction. I know it looks pretty long, but by the end, I promise you, you'll get every part of this. So like we mentioned, you already know half of this. So we said carbon dioxide. We need the carbon atom from this. We need the nitrogen atom from ammonium. We need the nitrogen atom from aspartate, okay? In addition to that, we also have 3 molecules of ATP and then 2 molecules of water. And then of course, we have urea, right? That's gonna be the product of the urea cycle. We'll also end up with fumarate, which we'll talk about in a bit. But mainly, we need to produce urea so that we can excrete it in the urine. Okay, and so this is pretty much the entire cycle, and we'll go through it using this diagram. This is the diagram that you guys have in your slides, and I feel like it's a little easier to understand. So let's quickly go over it and then go through every step in detail. So first of all, we start with ammonium, bicarbonate, and 2 molecules of ATP. And the most important thing is that we're starting in the matrix, so within the mitochondria. So the first product that we're going to be making is carbamoyl phosphate. And to synthesize carbamoyl phosphate, we have carbamoyl phosphate synthetase 1. Now, my favorite thing about the urea cycle is that the enzymes are very easy to memorize because they're literally telling you what's happening in the reaction. So we're synthesizing carbamoyl phosphate, and to do that, we need carbamoyl phosphate synthetase 1. Now, another thing, as you can see here, there's another molecule, and this is an extremely important molecule. I think I'll mention it about 10,000 times in this lecture. And it's, if you want to take away one thing from the urea cycle lecture, it's this. So N-acetyl glutamate is the obligate activator of carbamoyl phosphate synthetase 1. What does that mean? It basically means that without N-acetyl glutamate, this reaction is not happening. Okay, so in order for carbamoyl phosphate synthetase 1 to be active and to produce carbamoyl phosphate, it depends on N-acetyl glutamate to activate it. Okay, so this is very important. Like, if you will definitely get a question about this, so just make sure you understand this concept. And I'll keep repeating it again. This is just like a brief introduction. So after we make carbamoyl phosphate, our next product is going to be citrulline. And so to make citrulline, we're going to combine carbamoyl phosphate with ornithine. And this is the enzyme over here, which is ornithine transcarbamylase. And then, as you can see, we move from the matrix to the cytoplasm. So citrulline moves to the cytoplasm. And I'll show you guys a really cool way to remember this later on. But now that citrulline is in the cytoplasm, we make argininosuccinate. And then argininosuccinate is broken down into arginine, and then arginine is also broken down, and that's the end of the cycle. So that's pretty much everything. We'll talk about a few details here and there. And yeah, okay. So this is a cool mnemonic that I found. If this doesn't work for you, there are so many other mnemonics on the internet, but I thought this was cool. So let's go through it together. Orange colored cats always ask for awesome umbrellas. And you'll see this really cool picture over here. I'll put it in almost every slide so you guys can remember. So let's go through it together, okay? Orange is for ornithine. C in colored is for carbamoyl phosphate. Cats, citrulline. Always aspartate. Ask for argininosuccinate. 4 is for fumarate. And then awesome is for arginine. And umbrellas is finally for urea. And you can see that over here. Okay, so these are the substrates that we talked about, or the reactants, intermediates. And then again, about the enzymes, we have carbamoyl phosphate synthetase 1, okay, which will synthesize our carbamoyl phosphate, right, which is over here, okay? And then ornithine transcarbamylase for the second step, and then argininosuccinate synthetase, and then argininosuccinase. Okay, so, okay, remind me guys, where does it all start? Where does the first step of the urea cycle start? Or what does this, what is this arrow pointing to?
The matrix of the mitochondria.
Perfect, the matrix. So here you'll have 2 steps of the urea cycle, the first 2 steps. The first one to make carbamoyl phosphate, right? And then the second one to make citrulline. So these 2 products are made here in the matrix of the mitochondria. Okay, so with that, let's talk about the first step. And I have a little diagram over here to help you like orient yourselves as to where you guys are. So, okay, so like we said, we have ammonium, we have bicarbonate and 2 molecules of ATP, and we're synthesizing carbamoyl phosphate. So obviously the enzyme is going to be carbamoyl phosphate synthetase 1. Now, like we mentioned, in order for carbamoyl phosphate synthetase 1 to work, we need the obligate activator. And this obligate activator is N-acetyl glutamate. So again, in order for this reaction to move forward, in order for us to produce carbamoyl phosphate, we have to have N-acetylglutamate, or else carbamoyl phosphate synthetase won't work. And to put this into kind of like a picture format, here's something I created. Hopefully it helps in some way. So here, this is what carbamoyl phosphate synthetase 1 would look like without its obligate activator, right? Can't do anything. The reaction's not going to happen. You won't have carbamoyl phosphate. But then you add the obligate activator. So we have carbamoyl phosphate synthetase 1 with N-acetylglutamate. And finally, your reaction can proceed. Okay, so remember, this enzyme does not work without N-acetylglutamate. In other words, without N-acetylglutamate, the first step of the urea cycle can't occur. So you won't have the entire urea cycle. Okay, does this make sense to you guys? Because this is a very important point. I'll keep repeating it again, but just to make sure.
Yeah, that's cool.
Okay. Tamam, so we're here right now. So you'll see this after every step, just to show you guys how much we've covered. Again, this is to help you remember the mnemonic. So now let's move on to the next step, which is making citrulline. And again, we're still in the matrix, okay? So in order to make citrulline, we have our carbamoyl phosphate, which we made in the last step, the first step. We combine it with ornithine and we get citrulline. And now the enzyme for this is, the name's a little long, but I'll break it down for you guys and help you understand where it comes from. And it's called ornithine transcarbamylase. So let's look at why it's named that way. So transcarbamylase basically means that we're transferring a carbamoyl group, okay? And where this carbamoyl group comes from is from carbamoyl phosphate. So we're taking this part over here and we're adding it to ornithine. And this is what we get. So this is the carbamoyl group, and then this is ornithine, okay? And that makes citrulline, which is why the enzyme is called ornithine transcarbamylase, because we're taking the carbamoyl group and we're transferring it to ornithine. And in the process, you have a high energy phosphoanhydride bond that's being broken down. But I kind of explained the reaction just so you could remember the enzyme name. You don't have to know specifically the groups in detail and the way that I explained it, but just to help you remember the enzyme. Okay. So like I said, citrulline goes into the cytoplasm, right? So the first two steps, making carbamoyl phosphate and making citrulline, happens in the matrix. And then I kind of made up this sentence for you guys, which is citrulline sits in the cytoplasm. It kind of rolls off your tongue and helps you remember that citrulline goes to the cytoplasm. And so let's quickly recap on the steps so far. So first of all, we have ammonium, we have bicarbonate, we have 2 ATP molecules. And this makes carbamoyl phosphate. And the enzyme is carbamoyl phosphate synthetase 1. And we have N-acetylglutamate, which is the obligate activator of this enzyme, meaning without this, the enzyme won't work. And then carbamoyl phosphate combines with ornithine, right? We're transferring the carbamoyl group, so it's called ornithine transcarbamylase, and we get citrulline. And so citrulline sits in the cytoplasm, so it gets out of the matrix. As you can see here, it goes from the matrix and goes into the cytoplasm. So any questions on, well, you basically did 50% of the cycle. Any questions so far?
Yeah, does citrulline, when it goes to the cytoplasm, does it connect to the ortholine before it goes to the cytoplasm or after?
So that's a good question. It connects with it before. Okay, so here you have carbamoyl phosphate, right? So carbamoyl phosphate with ornithine makes citrulline. Okay, and then citrulline, once it's produced, it goes into the cytoplasm. So ornithine is used in the reaction to make citrulline. You know, it doesn't combine with citrulline itself.
Okay.
Does that make sense?
Yeah.
Okay. Okay, so let's start now with citrulline. Let's see what happens to citrulline in the cytoplasm. Okay, so this is where we are. And so citrulline is going to combine with aspartate. And in this reaction, we're using a molecule of ATP. So we're breaking down one ATP molecule, and we end up with arginine succinate. Okay, and because we are synthesizing argininosuccinate, then the enzyme is going to be argininosuccinate synthetase. Just remember that this reaction utilizes a molecule of ATP, so it's dependent on ATP. Okay, so just to repeat that step, we have citrulline combines with aspartate. Using a molecule of ATP, we form argininosuccinate, and the enzyme is called argininosuccinate synthetase. And so after we form the argininosuccinate, we actually just want to break it down, okay? So we form argininosuccinate, and then we're going to break it down. And to break down our argininosuccinates, our enzyme is called argininosuccinase because it's breaking down argininosuccinate. However, another name for it that you might find in questions is argininosuccinate lyase. And they're both basically the same enzyme. So I put both names for you here just in case. And so when we're breaking this big molecule down, it's argininosuccinate. It's going to give us two molecules. It gives us arginine, which is this over here, and fumarate. Okay, so what happens is, so again, we're breaking down argininosuccinates by argininosuccinate lyase or argininosuccinase. And so when we break it down, we get two substances. We get fumarate and we get arginine. So fumarate will leave the cycle and arginine continues, okay? And just like we broke down argininosuccinate, we also want to break down arginine. And so that's the final reaction of the urea cycle. So we're breaking down arginine, the enzyme is arginase, and we get ornithine, and we also get urea. So once you see urea, you know that you're at the end, right? However, ornithine, do you guys remember what step we saw ornithine in?
Yeah, it connects with something to form the other C. I'm not sure what it's called.
Yeah,
To
exactly.
get out of the cytoplasm.
Yeah, perfect. Okay, so ornithine, we saw ornithine in the second step. So ornithine combines with carbomoyl phosphate and it makes citrulline. So the arginine, when arginine is broken down, right, we get two things. We get urea, which is excreted in urine, and we also get ornithine. And because this ornithine is actually needed in the second step, it's needed in the matrix. So what's going to happen is the ornithine will go from the cytoplasm and go back into the mitochondrial matrix and enter this reaction again. So it's going to be used within the cycle. Does that make sense?
Yeah.
Okay. And the enzyme again is arginase. Okay, so like we said, ornithine is going to enter the matrix and combine with carbomoyl phosphate to make citrulline, and then citrulline goes into the cytoplasm, and then the urea is just going to be excreted. Now, there's this point that was mentioned in your slide, so I'm going to mention it here just in case. It basically means, so let me read it for you. There is no net loss or gain of ornithine, citrulline, argininosuccinate, or arginine, which means that we're not making more or not making less. So the levels of them are staying the same. So we're not gaining any of these molecules or we're not losing any of them. Just a point to keep in mind. So quickly, let's go over them again. So we start at the beginning, we start in the matrix, right? We have ammonium, bicarbonate, and 2 ATP molecules, and we use these three to make carbomoyl phosphate, and the enzyme is carbomoyl phosphate synthetase 1. And so N-acetyl glutamate is the obligate activator of this enzyme, meaning without it, it's not activated and the reaction doesn't take place. And now after we get carbomoyl phosphate with ornithine is going to make citrulline. And the enzyme here is ornithine transcarbamylase. Once we get citrulline, we said that citrulline sits in the cytoplasm, so it will move into the cytoplasm. And then we have our third reaction where citrulline combines with aspartate with one molecule of ATP, and we get argininosuccinate. And because we are synthesizing argininosuccinate, the enzyme is called argininosuccinate synthetase. And so once we make our argininosuccinate, we will break it down. And the enzyme is argininosuccinate lyase or argininosuccinase. And when we break down argininosuccinate, we get two substances. We get arginine and we get fumarate. And we said fumarate is going to leave the cycle. We'll see what happens to it later. And arginine is also going to be broken down. So this gives us the last reaction of the urea cycle. So arginine is broken down by arginase, and you get urea, which is excreted in urine. And then you also get ornithine, which goes back into the matrix and combines with carbamoyl phosphate to make citrulline. I know it's a lot of repetition, but this is the way that you guys will end up memorizing it, is just by repeating it, even when you're studying on your own. So any questions about the cycle? I'm open to any question. No questions? Okay. I guess silence is an answer, so. Okay, so I have a question for you guys, actually. We kept mentioning fumarate, right? We said arginine or succinate is broken down into arginine and fumarate. So what cycle or what molecule over here in the urea cycle is also a part of the Krebs cycle, which you guys took earlier on? Does anyone remember?
I didn't study the other cycle, but I think it's arginine.
So you're close. It's kind of fumarate. So it's in the same reaction, but it's fumarate. So fumarate is a part of the citric acid cycle. And so what happens is fumarate is a part of the citric acid cycle, like we mentioned. And because it's a product of the urea cycle, so let's just go back quickly. So, okay, I can't move the slides, but we can just use this diagram. So we said when we break down arginine or succinate, we get arginine and we get fumarate. And so fumarate enters the citric acid cycle, where it's converted into malate and then into oxaloacetate and then back into aspartate. And then aspartate can enter again into the urea cycle, where it combines with citrulline to make arginine or succinate. So this is just a way of showing you guys that these two cycles are connected and they're related in some way. Okay, so again, when we break down arginine or succinate, we get arginine, we get fumarate, right? So fumarate enters the citric acid cycle, and then our final product is going to be aspartate, which can then enter the urea cycle again, where it combines with citrulline to make arginine or succinate. And because we are synthesizing arginine or succinate, if you guys remember, the enzyme for this step is arginine or succinate synthetase. Okay, so just to repeat a few more points that we already mentioned. So we said that the urea cycle is there to remove ammonia, right? Because ammonia can be toxic when it builds up in our blood. So the body has to have a mechanism in which we can remove it, and that's why we have the urea cycle. Now, the overall control of the cycle is done by the enzyme levels. What does that mean? So whenever we have higher enzyme levels, we're going to have an increased rate of the urea cycle. So it's going to be working more than it would be if the enzymes were lower, right? Because the enzymes are what driving the reactions, and vice versa. Another thing is that the urea cycle also depends on the levels of the intermediates, so such as carbamoyl phosphate, citrulline, arginosuccinate, arginine, and all of those. So if those levels or if those intermediates are high, then you'll have an increased flow of the urea cycle. And if those intermediates are low, then the urea cycle won't be able to proceed, right? Because it depends on these intermediates and also the enzyme levels. Now, one of the most important points, which I already mentioned a few times, is the first step. And this first step of the urea cycle, where you're making carbamoyl phosphate, this is called the critical step. And so the way that we can regulate, or the way that the body regulates the urea cycle, is through carbamoyl phosphate synthetase 1, which is the enzyme of the first reaction. And so this is the reaction again. So we said we have a molecule of ammonium, we have bicarbonate, we have 2 molecules of ATP, and by carbamoyl phosphate synthetase 1, we get carbamoyl phosphate. And again, the obligate activator is N-acetyl glutamate. So this reaction has a lot of things that it needs in order to proceed, which is why it's the critical step. It's how we can regulate the cycle. So N-acetyl glutamate is so important, and you guys know most of it already, but there are a few more details that we should touch on, which is why this step is known as the critical step, right? So there's a lot of details that go into it. Okay, so N-acetyl glutamate, right? Again, the obligate activator of carbamoyl phosphate synthetase 1. So from the name, you can see that it's made up of two molecules, right? The acetyl part and the glutamate part. So first you have acetyl-CoA, and that combines with glutamate to make N-acetyl glutamate. And so because we're synthesizing N-acetyl glutamate, then the enzyme is also called N-acetyl glutamate synthetase. Now, the twist is that this enzyme relies on another molecule in order to be active or in order to work, and that's arginine. So in summary, in order for us to have N-acetyl glutamate, it kind of depends on two things. It depends on whether we have high levels of acetyl-CoA and glutamate, but also whether we have high levels of arginine, because N-acetyl glutamate synthetase, in order to make N-acetyl glutamate, requires arginine. So this is how we make the obligate activator of carbamoyl phosphate synthetase 1. And this part over here, the fact that arginine is needed for N-acetyl glutamate synthetase, this is an important point. It could come as an MCQ, so just make sure that you understand it. Again, we can go over it, we can repeat it again. So to make N-acetyl glutamate, you have acetyl-CoA and you have glutamate, right? The two parts of the final product. And to synthesize it, the enzyme is called N-acetyl glutamate synthetase. And this enzyme relies on arginine. So you could get an MCQ that's like, what product relies on arginine to be synthesized, or something like that. And so you would choose an acetyl glutamate. Does that make sense? Any questions?
No, can you just repeat the way the MCQ could come?
Okay, so I actually have an example for you guys at the end. It's a similar question, but they could simply mention something like, there's a product that's involved in the urea cycle in some way that requires arginine in order to be synthesized. Something like that. It won't be exactly word for word, but it's the concept that they're asking you about. So they want you to know that in order for us to have an acetyl glutamate, we require arginine, because arginine is going to activate the enzyme.
Cool.
Makes sense?
Yeah.
Okay. There's a question at the end, so you'll get a feel of how it would be like. Okay, so we're done with mostly everything. We have the genetic deficiencies and practice questions left. Should I continue, or do you guys want to take a break? It shouldn't take long, honestly.
I don't think we need a break.
Okay. Okay, so genetic defects, right? Let's start with ornithine transcarbamylase. So this could also come as a question for you guys, where they ask you, what's the most common disorder of urea deficiency? And that's OTC, which is the ornithine transcarbamylase. So we'll talk about four. We'll talk about ornithine transcarbamylase deficiency, carbamoyl phosphate synthetase 1 deficiency, which is this first reaction over here. We'll talk about what happens in case of ammonium toxicity and excess glutamine, and also argininosuccinase deficiency, which affects this reaction over here. So we wouldn't be able to break down argininosuccinate into arginine. But again, I know it sounds simple, but this could be an MCQ where they ask you which disorder is the most common. And so you'd pick ornithine transcarbamylase. Okay, so with that, let's start with the most common disorder, which is ornithine transcarbamylase deficiency. So whenever you have a urea cycle defect, so whenever you have an enzyme that's deficient, the question will always mention three main things. And one, that's elevated levels of ammonia, which is hyperammonemia. Since the urea cycle is the body's way of getting rid of ammonia, If the urea cycle doesn't work, ammonia will accumulate. And that's why we have elevated levels of it. Another thing is that you'll also have elevated levels of glutamine. If you guys remember, we said that glutamine is the way that the body can transport ammonia to the liver so it can be incorporated into the urea cycle. And so if we have a lot of levels of ammonium, then these are going to be converted into glutamine as well. So you'll have high levels of both. And another thing is you'll have decreased blood urea nitrogen. And that's easy to understand because you have the urea cycle, right? And the whole purpose of the urea cycle is to produce urea. And if the urea cycle isn't working, then we don't have urea. So that's why urea would be decreased. So whenever you see these three things in a question, you immediately know that you're dealing with a urea cycle question or a genetic deficiency in the urea cycle. So how do these babies present? So this is usually in the postnatal period. So after they're born, during the first 24 to 72 hours, they start to have symptoms of lethargy, vomiting. They could be hyperventilating. And obviously, if this is not treated, then it could progress to a coma or death as well. Now, what happens if ornithine transcarbamylase is deficient, right? So in general, the deficiency of CPS1, which is carbamoyl phosphate synthetase 1, and OTC are pretty similar, but there's one main thing that differentiates them. And that's what we're going to focus on because that's what you'll get in the question. And whenever you see this one keyword, you'll know to pick ornithine transcarbamylase immediately. And that's an increase in orotic acid. So it's only in ornithine transcarbamylase deficiencies that the patient presents with increased levels of orotic acid in the urine. So it's called orotic aciduria. And why does this happen in an OTC deficiency? And why doesn't it not happen in a carbamoyl phosphate synthetase 1 deficiency? And that's because when we don't have ornithine transcarbamylase, we won't be able to convert carbamoyl phosphate into citrulline, right? So what happens is carbamoyl phosphate is going to accumulate, okay? And when carbamoyl phosphate accumulates, it's going to start entering other pathways, other reactions, because the body wants to get rid of something that's accumulating, right? So instead of progressing through the urea cycle, which it can't do anymore because we don't have the enzyme, it's going to go into the pyrimidine pathway. So the pyrimidine pathway is where we can synthesize our cytosines, the thymines, and the uracil. And so what happens is this pyrimidine pathway is what causes increased levels of orotic acid. And this only happens in ornithine transcarbamylase deficiencies. So let's say we had a deficiency of this enzyme, carbamoyl phosphate synthetase 1. The reason why we don't have orotic acid urea is because we can't make carbamoyl phosphate in the first place. And so carbamoyl phosphate isn't being made, so it doesn't enter the pyrimidine pathway and you don't have any orotic acid. However, in an ornithine transcarbamylase deficiency, carbamoyl phosphate accumulates, it enters the pyrimidine pathway, and you'll find elevated orotic acid in the person's urine. So if you find this in a question, any question that says the person has elevated levels of orotic acid or increased levels of orotic acid or orotic aciduria, then you definitely just go for ornithine transcarbamylase deficiency. It's the only one that will present with that in this case. Okay, and so for treatment, usually for urea cycle deficits or defects, we give a low protein diet. And that's because ammonia actually comes from protein. So whenever you have a meal that's high in protein, your body is going to break down that protein, right? And this protein is broken down into amino acids. And these amino acids are also broken down into ammonia. And so you have elevated levels of ammonia. And so whenever you have more protein, the urea cycle needs to work a lot more to get rid of the ammonia and secrete it or excrete it in the form of urea. And so if the urea cycle isn't working, then we can decrease the protein that this person is taking so that, you know, the urea cycle doesn't have to work as hard to get rid of ammonia. Does that make sense? So that's why we just give a low protein diet. Make sense? Any questions?
Not really, because I didn't get it so well. The last part.
The protein part, right?
Yeah.
Tamam. Okay. So let's say I have a meal that's high in protein, right? Um, it's going to go through my digestive system. It's going to be broken down into amino acids, but it'll also cause an increased level of ammonia. So whenever we have a protein meal, this protein will cause increased levels of ammonia. Okay, so usually if a person has no genetic deficiency of the urea cycle, then the urea cycle can get rid of all this ammonia and excrete it in the form of urea, so you're fine. However, the problem is if a person has, let's say, an ornithine transcarbamylase deficiency, right? And they're eating a lot of protein, they're going to have really high levels of ammonia, but the body doesn't know how to get rid of it because the urea cycle isn't working, right? So in terms of treatment, how we can help these people is that we can tell them to eat less protein so that there would be less ammonia that the body has to get rid of, because the way in which the body gets rid of this ammonium, that way isn't working, right? The urea cycle isn't working because we're missing an enzyme. So that's why we give them a low protein diet. Was that better?
Yeah, that's clear.
Okay. So another thing that you'll see is an elevated level of glycine, glutamine, and alanine. So these three will also build up when it comes to either carbamoyl phosphate synthetase deficiency or ornithine transcarbamylase deficiency. Okay, so this is just a quick representation of the ornithine transcarbamylase deficiency. So this enzyme is deficient, right? So carbamoyl phosphate won't be made into citrulline. Remember the second step where we said carbamoyl phosphate with ornithine makes citrulline. So this can't happen anymore. So carbamoyl phosphate accumulates, and then it enters a specific pathway, which is the pyrimidine pathway, and then we get elevated levels of orotic acid. So that's the main factor that differentiates an ornithine transcarbamylase deficiency with a carbamoyl phosphate synthetase 1 deficiency, which is this enzyme over here. Okay, and this is a table you have in your slides. So as you can see, everything is pretty much the same except for the orotic aciduria. And that's going to be the way that the question presents itself, right? It's either no orotic aciduria or you will have orotic aciduria and you'll be able to say that, okay, this is ornithine transcarbamylase. So this is your keyword. I cannot emphasize this enough. This is really important and it's really going to help you when you're solving MCQs. Okay, so we said that we have elevated levels of glycine as well as glutamine, right? And so if something is accumulating in the body, then we need to give that person specific form of treatment that allows us to get rid of that, right? So if we have elevated levels of glycine, we can give something known as benzoate. And what happens is that this benzoate gets converted into benzoyl-CoA, and it can combine with glycine, and it makes hippurate. And the special thing about hippurate is that it can actually be excreted. So we're trying to get rid of it, right? So for glycine, we give benzoate, again, which can combine with glycine, and then it can be excreted in the form of hippurate. And then for glutamine, it's the same concept, but instead of using benzoate, we give phenyl acetate. So phenyl acetate is converted into phenyl acetyl-CoA. It combines with glutamine, and then we get phenyl acetylglutamine, which can be excreted. So this is the way that we can deal with the elevated levels of glycine and glutamine. And so the reason why we can't just leave the person or let the person be with ammonium toxicity is because whenever you have elevated levels of ammonium, or actually elevated levels of glutamine and glutamate, this can actually lead to brain damage, and it can cause edema, right? So swelling of the brain, because it absorbs water. So we can't have that. We have to stop that from happening. And the way that we can do that is by decreasing their levels by giving benzoate and phenyl acetate. So just try to remember that benzoate is for glycine and phenyl acetate is for glutamine. And finally, last deficiency is the arginine deficiency. And so arginine is this enzyme over here, also known as arginine lyase. So the reaction is meant to break down arginine to give us arginine and to give us fumarate. We said arginine will continue, right? It's going to continue in the final reaction of the urea cycle, whereas fumarate is going to leave the urea cycle and go into the citric acid cycle. So what happens if we don't have arginosuccinate lyase? So the enzyme that breaks down arginine is deficient. We don't have that, right? So if we can't break arginosuccinate down, then it's going to accumulate. And so people that have this deficiency will present with argininosuccinate in the urine. So instead of secreting urea, which they can't make, right, they're going to be excreting argininosuccinate in their urine instead. Which is what you see here. So the result is that argininosuccinate is secreted in place of urea. And it's going to be accumulated because the enzyme that breaks it down is deficient. And so for these people, you also want to give a low protein diet. If you have a low protein diet, you'll have less ammonia. So the urea cycle doesn't have to work as much. That's one way to think of it. And the reason why we give a diet that's also high in arginine is, if you remember, do you remember what arginine stimulates? Is it too soon to ask that question? But our arginine basically stimulates an N-acetylglutamate synthetase, which we said was the enzyme that we needed to make N-acetylglutamate, which is the obligate activator of carbamoyl phosphate synthetase 1. Okay, so that will basically help drive the reaction forward and maintain the urea cycle so that we can still get rid of our ammonia. Because again, if it accumulates, it's going to be toxic. Okay, and with that, we're done with the lecture. If you guys have any questions, well, there's like one person here, but if you have any question, I can answer it now, and then we can go through the practice questions.
Can you go 2 steps back, like 2 slides,
Tamam.
to the second deficiency?
Was it this one?
No, the next one, I think.
Next one? This one?
Yeah, that one.
Okay.
So you said if there is arginine succinase deficiency, we will won't be able to produce arginine, so we won't be able to produce urea and arginase and orthanine. Sorry, not arginase, orthanine. So how do we get the more orthanine to produce the citrulline and then proceed the process?
That's a very good question. So as I mentioned that we give a diet that's high in arginine as well, right? And you actually reminded me to mention something else, so thank you. So this arginine not only is going to be used to increase the levels of the obligate activator, right, by activating the enzyme, but it can also enter the cycle through this step, right? So arginine can also come in through here, and then it can produce our ornithine and our urea. And so the ornithine can go back into the matrix and still be used within the cycle. So arginine will have you can say that it has 2 benefits, right? One is to increase the production of N-acetylglutamate, and then the other one will be to just simply enter the cycle at this point, right? It's kind of like you're bypassing the step because you can't have the step. So instead of relying on argininosuccinate to give us the arginine that we need, the arginine can come in from the diet and then just enter the cycle from here. And that's how we can get our ornithine.
Cool.
Makes sense?
Yeah.
Okay. Do you want to do questions?
Yeah, of course.
Okay. So, okay, you want to read the question on your own, or? Yeah, I think I'll give you time to read the question and then we can explain it and discuss it together.
Glutamine or glutamate. It starts with glut.
Okay, it starts with glut. You were right the first time.
Glutamine.
Yeah. So remember when we said that glutamine is the non-toxic carrier of ammonia? So
Yeah.
D is going to be the answer. So this is a fairly simple question. This should be like a recall question, you know? So I tried to combine a bit of both different styles of questions so you guys could get the hang of it. Okay, and this is the slide over here. Okay, next question.
V,
Again?
B,
Okay, so let's go over the question. So tell me why you said B. Why did you choose ornithine?
because orotic acid urea.
Okay, so we said whenever we have orotic acid urea, it's because which enzyme is deficient?
The ornithine
The
synthase, something like that.
Yeah, ornithine transcarbamylase. And ornithine transcarbamylase, can you remind me of the reaction? Do you remember it?
I need a sec, wait.
No worries. I can mention it again, it's fine.
It's the last reaction, like the one they.
Go back, wait, it's this one. So here, so we said carbamoyl phosphate with ornithine gives us citrulline, right? And the enzyme is ornithine transcarbamylase. So if we don't have ornithine transcarbamylase, we won't be able to make citrulline, right? So we have ornithine and we have carbamoyl phosphate. But in order for us to make citrulline out of these two substances, we need the enzyme. And so if the person has an ornithine transcarbamylase deficiency, then we can also expect that they won't have citrulline. So the answer to the question should be C. Does that make sense? Because ornithine is used to make citrulline. So by ornithine transcarbamylase. So the enzyme combines ornithine with carbamoyl phosphate and it makes citrulline. So if we don't have the enzyme, then we won't be able to make citrulline.
Yeah.
Make sense? I can explain.
No,
Okay.
it did make sense. It just didn't read the second part.
Tamam, okay. Okay, so this is the pathway here if you want to revise it again. Okay, next question.
C.
Okay, let's go through the question together.
Or B, sorry, B. If I'm
Here, C.
no B. I changed
Okay.
my answer.
Right. We'll go through the question together. Okay, so we're talking about a newborn, right? And immediately we can see that they have high levels of ammonium, glutamate, and alanine. So we know that this is a urea cycle genetic deficiency. So they performed a urinalysis, so they examined the urine, and they found that levels of orotic acid and citrulline were not elevated. So the person didn't have increased orotic acid and didn't have increased citrulline. So that means that the enzyme that's deficient has to come before citrulline is produced, right? So let's look at the diagram. We can use this pathway over here. Okay, so we said that there's no orotic acid urea, right? Which means that ornithine transcarbamylase is not deficient. We know that for sure. But however, the citrulline levels were also not high, right? So if it's not ornithine transcarbamylase, then it's probably going to be carbamoyl phosphate synthetase. So let's say arginosuccinate synthetase or arginosuccinate lyase were deficient, then we would have elevated levels of citrulline, right? Because the urea cycle can't progress. It's kind of stuck at this step. So if the enzyme arginosuccinate synthetase, which takes citrulline, combines it with aspartate to make arginosuccinate, if this enzyme is deficient, then citrulline is going to start to accumulate. right? So if the question says that citrulline levels are not elevated, right, that means that the enzyme that's deficient should come before citrulline. And because there's no orotic acid urea, then that tells us it's carbamoyl phosphate synthetase 1. Wait, let's go back to the question. Yeah. So again, let's look at the question. So the main keyword here is when they examined the levels of orotic acid and citrulline, both levels were not elevated. And so if it was argininosuccinate lyase or argininosuccinate synthetase, citrulline is already produced before that step. So citrulline would probably be elevated in that kind of deficiency. But in the case of carbamoyl phosphate synthetase 1, if this enzyme is deficient, then we won't be able to make citrulline. So it would make sense for citrulline not to be elevated. Was that clear?
Yeah.
Okay. Fine. Next question. Let me help you. This is a very big keyword over here. Increased orotic acid.
It's E.
Perfect, it's E. So this is a question that could definitely come. So and as soon as you see increased orotic acid, then it's 100% ornithine transcarbamylase. Okay, good job, guys. Okay, and this is the table again. So orotic aciduria only happens in ornithine transcarbamylase deficiencies. Okay, next question. Okay. Anyone have an answer? I'll give you a hint. So it's asking, so the ability of arginine to render this effect stems from its role in activating the synthesis of an activator of one of the enzymes of the urea cycle. So long story short, it's basically asking.
I know the answer. It's E.
Perfect, it's E. Okay, so why is it E? Remember when we
Because
said
arginine activates the N-acetylglutamate, which activates the other enzyme.
exactly. So we need arginine for the enzyme that makes an acetylglutamate. And so I know it's a long question, but at the end, you literally only need this one line. So it says, which of the following represents the potent activator whose synthesis is induced by arginine? So usually the question is going to be the last line, but you might need some like buzzwords in the middle of the, like in between the lines. But for this, it's just the final sentence. And so you guys are absolutely correct. It's an acetylglutamate. And this is the slide over here. So we said arginine is needed to activate N-acetylglutamate synthetase, and this synthesizes an acetylglutamate. So without arginine, we won't be able to have our obligate activator. Okay, next question.
This one is B.
Okay, So read this part over here. It says we have elevated levels of citrulline, right? But we don't have elevated levels of argininosuccinate. So argininosuccinate is not elevated. If B was deficient,
So
then
C, right?
yeah, it's C.
Yeah, because it's before the arginine comes.
So if B was the answer, then they would probably mention in the question that argininosuccinate is elevated. So yeah, you guys in the chat, C is the correct answer. So why? Because if we don't have the enzyme that synthesizes argininosuccinate, then we won't have argininosuccinate. So its levels won't be elevated. And then another thing where it says that we have a marked elevation of citrulline, that also makes sense because if you see the pathway over here, so we said that argininosuccinate synthetase is deficient, right? This enzyme isn't working. So not only can we not make argininosuccinate, but citrulline is also going to start accumulating. So the person will have an elevated level of citrulline because the cycle can't progress forward. So they'll have elevated levels of citrulline, but also argininosuccinate levels won't be elevated. And so that will let you know that this is the enzyme that's deficient. Any questions? Okay. We can move to the next question.
B, protein.
Yeah, you guys are right. It's protein. And so we said that usually the treatment for urea cycle deficiencies or enzyme deficiencies is going to be a low protein diet because the outcome would be less ammonia. And so the urea cycle won't have a lot of ammonia that it has to excrete since it's not working as efficiently. Okay, so B, as you guys said. Okay, I think this is the final question. So you guys want to read it?
I think it's D.
You think it's so this reaction?
Yeah.
Okay, let's read the question together. So an infant born to a 32-year-old female demonstrates lethargy, vomiting, and seizures after her first few feedings. So by this, we know that it's obviously a urea cycle deficiency. Initial laboratory evaluation demonstrates a markedly increased blood ammonium level. So we have high levels of ammonium because we can't get rid of it, or the body can't get rid of it. And then liver biopsy suggests impaired formation of N-acetylglutamate. Okay, so if we can't make N-acetylglutamate, then there's a specific reaction in the urea cycle that won't work because N-acetylglutamate is the obligate activator of which enzyme? Do you guys remember?
Which enzyme? The citrate pro or something like that. The second reaction. I don't know what's called.
It's the carbamoyl phosphate synthetase 1.
Yeah.
Someone said A in the chat. A would be the correct answer. And that's because N-acetylglutamate, we can go back to the reaction over here. Yeah, so N-acetylglutamate is the obligate activator of carbamoyl phosphate synthetase 1. And so this person, when they did a liver biopsy, it showed that there was impaired formation. In other words, that the body couldn't make N-acetylglutamate. So our obligate activator is deficient, right? So this enzyme won't be active. And so the first step of the urea cycle won't proceed. So we won't be able to make carbamoyl phosphate. So let's go back to the question, and which is why the answer is A. So A is where we make carbamoyl phosphate. And so the enzyme carbamoyl phosphate synthetase 1 is involved in this reaction. And this is the enzyme that requires N-acetylglutamate. So if we don't have N-acetylglutamate, then the first reaction, so you can think of it, it's the most important reaction, it's the critical step, right? It requires an obligate activator, so it's the first reaction. So first, most important. So if N-acetylglutamate is impaired and we can't make it, then this reaction is going to be affected because we can't make carbamoyl phosphate. Make sense?
Yeah, but why D is wrong? Because I thought arginine was supposed to activate the N-acetyl glutamate. So then the carbonyl, I don't know what's called, carbonyl phosphate synthase won't be activated.
Carbamoyl phosphate. Okay, so you mean that, wait, can you repeat your question, please?
So I picked D because D is where arginine is produced, which activates N-acetyl glutamate. So if N-acetyl glutamate isn't activated, then carbamoyl phosphate synthetase 1 is not activated then.
Okay, I get what you're saying, but so the question is mainly asking about N-acetylglutamate. So there's impaired formation of it for whatever reason, whether it's the enzyme or whatever, but we don't have N-acetylglutamate. And so they're kind of hinting at which step is N-acetylglutamate directly activating. So that would be step 1. So arginine would be needed for the enzyme that makes N-acetylglutamate, right? But if we don't have N-acetylglutamate, then that affects the first step. Because N-acetylglutamate is not involved in this reaction over here. It's only involved in step 1. If that didn't make sense, we can go over it again.
It does, thank you. Just a question, where can we get the slides?
I can send it to your BLCs and they'll share it with you guys on announcements, inshallah.
Oh, okay, thank you.
I also included explanations in the notes and tips throughout the slides and summaries. So if you guys choose to go over this, then make sure to look at the notes section as well.
Thank you. I just have a question. Is that enough, or should we like use our doctor's slides?
So I always say make sure that your lectures are your first resource, no matter what. So study from the lecture first, and then if you want to go through this, then you can. I included everything that was mentioned in your lecture, so it should be fine. But again, just to be on the safe side, always use your lectures as your first resource.
Okay, thank you.
No problem.
Have a great day, doctor.
Thank you, you too. Okay, and that's it. Thank you.